if you have ever got a chance to visit some lab that deals with biotechnological aspects or some brewery even, you would have observed these huge constructions or arrangements inside the lab. Now what it is a bioreactor. We had said that during the downstream processing what happens is that once the recombinant protein is obtained, we make sure that the recombinant protein is obtained in abundant amounts, and it is then sent for packaging and then introduced into the market, and lots of profits are made out of it. And some have the aim of doing philanthropic activity, that human race gets rid of certain diseases. So where all this production takes place, that is the site known as bioreactor. This picture shows what a bioreactor looks like. There are two types of bioreactors that we have. Certainly this is a huge tank inside which you are going to have the nutrient medium supplied continuously. At the same time, because we are dealing with living cells, if they are aerobic, you have to supply the oxygen as well. You have to make sure that the conditions are sterile. So what are the conditions which you require? That aeration should be there. There should be completely sterile activities and continuous supply of nutrient medium. And lastly, it should be extraction of recombinant product, okay, or protein. These are the 4 steps which are aimed at, which one aims at achieving when he or she sets up a bioreactor. Now bioreactors are of 2 types. One is the stirred tank bioreactor, we have the simple representation of that, and the other one is sparged stirred tank bioreactor. So we are going to see both of them. There is a continuous agitation that takes place inside the bioreactor. So we have an agitator so that there is no stagnation, it keeps on mixing, the nutrients are used to their full availability. At the same time, we have a huge reactor tank inside which this agitation system is floating. Supposedly what you see over here or wherever you are sitting, that is an entire bioreactor. You have a huge tank, reactor tank, in which you find the nutrient medium. There are sensor probes which keep on checking whatever is being produced, okay, what is the quantity of the recombinant protein that is being produced, which are in contact with sensor monitor system monitors which keep a check on the production. The agitation system keeps on moving whatever is present in the reactor tank, and what is present in the reactor tank? It is the nutrient medium. There is an aerator, air supply inside it so that aerobic conditions are maintained, and the medium is continuously being added. The used up medium is given out from the effluent. So we have continuous supply being taken care of, there is aeration, and this reactor tank is completely closed, as you can see. So they have maintained the sterile conditions as well. There is a thermal jacket, often it is provided with water, okay, so that takes care of the temperature of the reactor that is to be maintained, that is given by thermal jacket. In certain diagrams, if you get to see, you will see that there is water layer that is present. That water acts as a coolant or acts as the buffer which is going to absorb the extra temperature. So this is a stirred tank bioreactor, the simplest form. In certain cases, there is another type of bioreactor, that is sparged tank, okay. Now it is quite similar to that thing only, but the difference is that there is a sparged air which is given up, okay, there is a air supply that is given up, and that sparger gives up air bubbles, okay. These air bubbles, air is given in the form of bubbles. Now those bubbles, they make the surface area increased and the reaction ability increases, and there is a headspace pressure, there are monitors which are observing what all is taking place, mixing takes place continuously. What you have to take care is that the air bubbles increase the surface area, and that are being added by a propeller or impeller which adds the sparged tank bubbles inside it. So this is the sparged stirred tank bioreactor that you have to remember. So we get to know that there are two types of bioreactors. In both the cases, the supply would be continuous. All these four conditions would be taken care of, and in one case you have aeration supply, in the other case you have sparged bubbles inside it, so that the surface area increases and the productivity of the bioreactor is many-fold. So this is all about bioreactors.